Fessel and Schmartingarten. Sorry, hold on, gotta take a bit of lip.
Time to learn about
Heredity.
It's the most important part of biology these days. You see, the DNA is arranged on chromosomes and segments that I like to call genes.
Not these kind of jeans.
These are like $20.
They are not. I bought them for $5 at Kmart.
The DNA in chromosomes is arranged in segments called genes. In a human body, there are about 46 chromosomes, which is a lot of DNA. And 23 are from your mom and 23 are from your old daddy, or you like to call pops. In a chromosome in the human body, one is from your dad and one is from your mom. And they mix together and they form on one chromosome, which you like to call a homologous chromosome.
A female gamete and a male gamete come together in a process called fertilization, in which they form a diploid, which is a zygote, or 2N. Gametes are formed during a process called meiosis.
In meiosis, it's kind of different than cell division that you all will know. In meiosis, the cell actually reduces the number of chromosomes that they have, which is kind of peculiar. Sure, what would you say?
Yeah, I would. Meiosis is split into 2 phases, meiosis 1 and meiosis 2, which is just like mitosis.
Wait. Mitosis?
Not those mitosis, you nitwad! Mitosis can be remembered by PMAT, prophase, metaphase, anaphase, and telophase.
Let me explain the rest. Hold my nanner.
Okay.
So, the difference in meiosis with regular cell division is that meiosis happens twice. Yeah, twice. But the chromosomes are reduced. You see, that happens because crossing over occurs. Crossing over is when 2 homologous chromosomes, you see we talked about those earlier, are changing and they exchange their chromosomatic structures and their genes are separated and their DNA are separated, so me and old Fessel over here don't look alike at all. See? It makes more sense now. Professor, why are you eating again?
I like my berries.
Well, since we're talking about these little things, let's just use them. So, there was an Austrian monk and a plant breeder. His name was Gregor Mendel.
Oh, I love that fella.
Of course you do. And he thought that one day we was going to use 2 different pea plants, one green and one yellow, but we're going to use blue today. And these 2 peas breeded together, and he thought that he was going to get some blue, some green, but no. He got all yellow, but we'll use blue. And he was thinking to himself, well, what happened? What did I do? And then that's when he found out that the yellow peas were
Dominant.
They were dominant, which means that they were the gene that overpowered the rest of the genes. Right?
Correct. Now let's talk to them about another word I like. When the gene gets masked over.
What? Here, you show.
Well, we said that the pea plants did not turn green. Well, that would mean that the green pea plants, or in this case berries, would be a word I like to call recessive.
Which happened in the second generation. In the second generation, Mendel found out that the peas weren't only yellow, but there was some blue in there too. But he only breeded 2 yellow ones, so how is that possible? But it turned out that the green was masked in the genes, which means that it was recessive. When Mendel explained his P's, he thought that he should explain it in a different way. So he thought that he should explain the genes in alleles, which are different forms, say, of a gene. And the genes could be represented by uppercase A, uppercase B, lowercase b, any letter that you wish. But the dominant allele would always have to be uppercase. And the recessive allele will always have to be lowercase. If you mix it up, then the whole shebang bang is messed up. Right?
Correct. Then, when an organism has two of the same traits, it would be homozygous. For this example, we have two blue peas, or as we've been saying, yellow peas. And then you come over here, and this parent, yeah, you heard right, it's heterozygous. One green, one blue, or yeller. And that is correct.
When Mendel tried to explain these allele pairs to other people, they didn't really get what it meant. So, Mendel tried to make up another word. So he called them genotype, which are the allele pairs. They could be uppercase A, lowercase b, or uppercase A, uppercase A. And the phenotype, which is the outer appearance side of the traits, like Professor's hair, or the way that Professor eats, or Professor's eyes. You see? We'll try to assort the allele pairs. It kind of got a little tricky. So he made up this different process we like to call. For example, this one is called a monohybrid cross. And let's put an example. In the dinosaur's chromosomes, there are two traits. One is for a blue skin and one is for purple skin. The mother is a heterozygous blue and the dad is a homozygous purple. If the blue skin is dominant, then what's the outcome for the child?
Well, you see here, we have the father staying on the top, which keeps him at a homozygous purple, since the lowercase b means purple. This is the mother being a heterozygous blue, and capital B for dominant blue, and that would be purple. Now you bring it over because we always want the capital B to be in front. So, big B, little b from up top, big B, little b from up top. Bring it over. In this case, it doesn't matter what order. Little b, then bring it over again, and from the top, little b, little b. You see, for the ratio here, it would be 1-to-1 or possibly 50%.
So that means that, anyways, there's a 50% chance that the dinosaur could either be purple or blue.
Mendel started working with more than one trait. He decided that a monohybrid cross would be too small for what he needed to work with. So, to fix this, he came up with a dihybrid cross.
You see, we're going to use the same example for this one about the dinosaurs, how the dad was homozygous purple, and then how the mom was heterozygous blue. But we're going to add a new trait, which is tall and short. The tall is dominant, so that's going to be represented by uppercase T, like you see right here. And the recessive is going to be represented by a lowercase t, which is right here. You see, the dad was a homozygous tall, so that's going to be represented by two uppercase t's. And the mom was represented by two lowercase t's, which is a homozygous short. You see, to make all of these workable for the dihybrid cross, you would, let's say, switch them over. You see, for one of these squares, you would put a little b and switch it with a big T. Same over here, you would use the same little b and cross over with the big T, like right here. And for the next one, you would use little b again and another big T, shown right here. And again, a little b with a big T, right here. Now for the mom, you would switch it with a little b a big B and a little t, sorry. And then for the next one, you would choose the same big B and choose another little t, as shown right here. Then, for the next one, you would switch to the little b and go to the little t, shown right here. And then for the last one, you would choose the little b again and go to the last little t, which is right here. Okay, now we're ready to start our dihybrid cross. Now you just fill it in, but remember, the uppercase always goes first. The uppercase right here is the big B. And since there's a big B right here, we're just gonna have to move on with the rest of the B's. So there's another lowercase b right here. Now let's move on with the T's. Remember, uppercase goes first. So there's a big T up here. And there's a little t right here. See? And it's just like that all the way down to the rest of the whole dihybrid cross. By the end, this is what your dihybrid cross should look like. All the spaces are filled in and all in the correct places. It's that easy. And maybe you might be able to find your own genes and traits. But it's pretty hard to find the ratio here, so I'm gonna have you do that on your own. And that's about all you need to know. From Punnett squares, to meiosis, to Mendelian genetics. Now you know everything about how you were born, or how you were made, or how your traits make you the way you are. See, it's that easy.